Welcome guys to my YouTube channel Real Science Simple, and let's make the science simple. I'm doing lectures on CRISPR, and today I'm going to cover the topic of base editing by CRISPR. So base editing could be done by two methods. One is the cytosine base editing or alanine base editing, or another one is the prime base editing. In case of the cytosine base editing, you can convert cytosine to uracil. In case of the adenine base editing, you can convert adenine to guanine. So here you can see the cytosine and uracil both are pyrimidine, so you can convert pyrimidine to pyrimidine, or you can convert, in case of the adenine base editing, you can convert adenine to guanine, which is adenine and guanine both are purines, so you can convert purines to purines. So this is the main drawback of cytosine or adenine base editing. In this technology of cytosine or adenine base editing, we can use two kind of Cas9 enzymes. One is the Dcas9, and the Dcas9 has a defective nucleus domain, and because of this they can recognize the PAM sequence, but they cannot cleave the DNA strand. Okay, so this is the Dcas9, and there is another Cas9 proteins which are known as the Cas9 nickase. As you know, the Cas9 has two kind of nucleus domain, which is one is the RUPC domain and another one is the HNS domain. RUPC domain can cleave the coding strand. Coding strand is also known as the non-template strand. It is also known as the non-complementary strand. Similarly, the HNS domain can cleave the non-coding strand. It can cleave the template strand, or it can cleave the complementary strand. Okay, complementary to the guide RNA. So if you do any mutation, not any mutation, but basically if you convert the histidine residues at 840 position to alanine residues, you are creating a mutation in this domain, and because of this it cannot cleave the non-coding strand, or you can say the template strand or the complementary strand. Similarly, if you are doing the mutation in the RUPC domain, which is basically you are converting aspartate residues at 10th position to alanine residues, then you cannot cleave the coding strand or the non-template strand or the non-complementary strand. Okay, so here you can see here basically what happened in this cytosine base editing. You are using the guide RNA, you are having the Cas9 protein which is fused with the cytidine deaminase. This cytidine deaminase will deaminate the cytosine residues and will convert this base pair, cytosine base pair, to the uracil. Now this cytidine and Cas9 protein is also fused with the UDG inhibitor, UDG inhibitor, which is the uracil D-glycosylase inhibitor. So what this inhibitor basically do? This UDG inhibitor will prevent the conversion of uracil to cytosine. So uracil cannot convert back to the cytosine residues if this UDG inhibitor is present. So this Cas9 protein is fused with the cytidine deaminase along with the UDG inhibitor. So what happened now? This cytosine will convert into the uracil, and because it is attached with the UDG inhibitor, this uracil will not convert back to the cytosine. Now the another thing, we forcefully want this particular unedited strand to be used as a template strand by the Cas9 protein. So we want to create a nick in this unedited strand, and for this region we can use the Cas9 nickase enzyme here. For example, which kind of Cas9 nickase you can use? RUPC, you want to do mutation in the RUPC domain, or you want to do mutation in the HNS domain. And this is basically, you can say, the coding strand, and this is, you can say, the non-coding strand. And as you know, the RUPC domain cleave the coding strand, and we do not want coding strand to be cleaved, so we will do the mutation in the RUPC domain, we will do the mutation in the Rupsi domain, where we will convert the aspartate residues at 10th position to A position. So which kind of Cas9 nickase we are going to use? We are going to use D10A Cas9 nickase here. So what it will do, the Cas9 nickase will not cleave this strand, but it will cleave this particular, the non-coding strand, and therefore this G, the mismatch repair enzymes, will convert this guanine to the adenine base pair, and therefore we can convert the pyrimidine to pyrimidine, which is the cytosine, to thymine in one strand, and to the another strand we are converting guanine to adenine. And the similar thing will happen with the adenine base editing, where we, instead of cytidine deaminase, we will be using the adenine deaminase. You can use any kind of cytidine deaminase here. And similarly, in case of the adenine base editing, where you want to convert adenine to guanine, you can use the adenine deaminase. Another editing which you can do by the prime editing. In the prime, the main difference between the prime editing and the cytosine or the adenine base editing is this, that in case of the cytosine or adenine base editing, you can either convert purine to purine or pyrimidine to pyrimidine. But what if you want to convert purine to pyrimidine? Then in that case you can use the prime editing method. In that you can use the prime editing guide RNA, which is also known as the PEG RNA, and that is fused with the Cas9 protein, which has the mutation in the HNS domain. So because it has the mutation in the domain, what it will do, HNS domain basically cleave the complementary strand. And if you are doing any mutation in the HNS domain, it will not cleave the complementary strand. It means this is your guide RNA, so this is going to be your complementary strand. Or complementary strand is also known as the non-coding strand and also known as the template strand. Okay, so it will not cleave this strand, and it will only cleave the non-complementary strand, and therefore we are using the specific kind of Cas9 nickase enzyme, which is the Cas9H840, which contain the mutation in the Cas9 protein as the 840 position, where histidine is converted into the alanine. Now it will only create a nick in the non-complementary strand, which is this strand. Let me highlight this, which is basically this strand. Okay, so now it will create a nick here, just 3 base pair upstream of the PAM site, and because it will create a nick, and therefore you will be getting the 3' flap region. Now please note this point, this guide RNA, which is also known as the prime edited guide RNA, which is the PEG RNA, has a specific sequence at the 3' position, and this sequence basically you have to design by yourself. Suppose if you want to convert TGA, which is the stop codon, to TGG, so that the stop codon in this particular region is going to be removed, then you need to create the sequence, which is like, if you want to convert TGA to TGG, so you need to have a sequence complementary to the TGG, which is going to be the ACC. Similarly, on the other sequences also you need to design as per your experiment. Now because it has a reverse transcriptase will convert this, or will produce the complementary sequence of this particular sequence, which is attached at the 3' region of the prime editing guide RNA. Now you have the 7 bases here, ACC, ACGT, which you have designed. So complementary sequence is going to be TGG, TGCA. Okay, now these 7 bases will replace the 7 bases here. So it will replace 1, 2, 3, 4, 5, 6 and 7. So it will leave only AA here, and this sequence is going to be cleaved by the FAN1 endonuclease. So now you have this sequence TGG in your non-complementary or the coding strand, TGC and AAA. So now you can see you have convert the TGA to TGG. So you have removed the stop codon here. You have also removed the PAM sequence here. So this Cas9 will not recognize any, because you know the PAM sequence is very essential for Cas9 to function. So Cas9 will not recognize any of these sequence here. Now the mismatch or repair enzymes will convert this sequence to this sequence. So where the TGA, which was the stop codon, has been converted into the TGG. So you can see the desired edit removes the stop codon, which is the TGA. It has removed TGA and it has converted TGA to TGG. Similarly, the PAM site is also being destroyed, and therefore the Cas9 protein will not cleave any of these sequences. So this is the modification that you have done. Suppose this is the modification that has not been done, then there will be the second attempt at the editing you would have to do in this experiment. Okay. So that's how the prime editing basically function, where you can convert the purine to pyrimidine, or pyrimidine to purine, or purine to purine, or the pyrimidine to pyrimidine. And the three main components here is the guide RNA, the Cas9 protein, Cas9 nickase, which has the mutation in the HNS domain, and therefore it will not cleave the complementary strand. It will only cleave the non-complementary strand. This is the complementary strand because this is complementary to the guide RNA. This is the non-complementary strand. So it will create the nick in the non-complementary strand, and because this guide RNA has the specific sequence that you will design according to your experiment. For example, if you want to convert TGA to TGG, so you need to have a sequence complementary to TGG or some other sequences. Now these 7 bases in this particular example will replace the 7 bases, and these 7 bases are going to be removed or by the FAN1 endonuclease. And since there is a conversion of the stop codon to TGG and the PAM site is also being destroyed, the mismatch repair enzymes will repair these DNA molecule, and that's how the prime editing basically function. So that's all about this lecture, and if you like the content of my lecture, kindly subscribe my channel, and thank you so much.